Good afternoon, everyone. Thank you so much for joining us today for our webinar on the research and science behind food allergy. I'm Lori Harada, the Executive Director of Food Allergy Canada, and I have the honor of introducing our guest speaker today, Dr. Manel Jordana, who is an MD, PhD, and Professor of Pathology and Molecular Medicine at McMaster University in Hamilton, Ontario. Dr. Jordana joined the Faculty of Health Sciences at McMaster University in 1988 and has since supervised more than 75 trainees and co-authored 160 scientific papers, reviews, and book chapters. With more news stories about research from around the world, we felt it would be beneficial for the lay public, people who don't have a medical or research background, to understand some basic concepts about what happens within the human body in our immune and other systems. So we asked Dr. Jordana to present today on the basic science behind food allergy research. As a scientist, Dr. Jordana does not have a clinical practice, so he does not see patients with food allergy. His research focuses on the immunobiology of allergic diseases, including asthma, as well as peanut allergy and anaphylaxis. Thank you to all of you who sent in questions before the webinar. We've received a lot. Many of them were about daily management of food allergy, the use of epinephrine to treat allergic reactions, and specific questions about the new early infant feeding guidelines. Given the scope of this presentation and the time available today, Dr. Jordana will not be covering these topics. We encourage you to visit our website, foodallergycanada.ca, where you can find more information. You can also watch our other recorded webinars, like the one on early infant feeding guidelines. There's one for the public, given by Dr. Julia Upton, and one for healthcare professionals, delivered by Dr. Edmund Chan. If you have specific questions about yourself or your child, kindly refer back to your healthcare professional. Some housekeeping items. Before we get started, there are a few. One, all of the participants on today's call are muted so that we can keep the audio clear for the webinar. Secondly, we'll be answering questions after the presentation, so feel free to submit them in the chat or question box throughout the webinar. We'll try to get as many questions as we can covered. And finally, this webinar will be recorded and shared on our site afterwards, so we encourage you to go back and watch it again and share the link to the webinar with your friends and family and colleagues. Now, it gives me great pleasure to introduce you to Dr. Manel Jordana.
Thanks very much, Lori, for this introduction. And thank you to all of you attending this seminar and the many, many interesting questions that you have submitted already. I'm hoping that in the course of this presentation, I will be able to answer at least some of them. This is the agenda for today's seminar. First, I will do a brief review of the allergic reaction, providing some insights, basic insights into the cells and molecules that participate in this reaction. Then we'll go on to talk about the before and after of an allergic reaction to foods. What do we mean by this? Well, the before, we mean the process of becoming allergic. And by after, we mean what may happen after you may have had an allergic reaction, which is persistence of the allergy. At the end, we'll discuss treatments, current treatments, and potential future treatments and their impact at different stages of the process. And we'll finish with a summary of the principal concepts. Let us begin with the brief review of the allergic reaction. And I like to focus on this word, immunoglobulins. Immunoglobulins are proteins that we produce and play a very important role in defense against viruses, against bacteria. There are 5 classes of immunoglobulins. IG for immunoglobulin, A, M, D, G, and E. We are particularly interested in IgE, and this immunoglobulin is important for defense against parasites, but also is the key immunoglobulin in allergy. Let's go to the heart of the matter of an allergic reaction. Here on the left, you can see an image of IgE, and it's this type of representation. So this molecule is able to bind to specific receptors on the surface of some particular cells. On the right, you can see the other key component of an allergic reaction is what we call the allergic cell, or also a mast cell. Here you can see a cross-section through electron microscopy of a mast cell. And notice that the unique and impressive characteristic of this cell is the presence of all of those granules inside. And these granules contain a wide variety of molecules. There is another representation now of a mast cell with all of these granules inside, and IgE molecules that are bound to the surface of this mast cell. When an allergen that is specific for this IgE molecule gets into contact, it initiates a series of reactions that are referred to as degranulation. And degranulation, basically what it means is that there is this fantastic explosion of D cell and the release of a wide array of molecules. Here's a list of these molecules, and you may recognize some of them. For example, histamine. Some of those molecules were contained within those granules, but there are other molecules such as leukotrienes and prostaglandins that are synthesized, but are synthesized within minutes. Collectively, all of these molecules mediate the signs and symptoms of an allergic reaction, from sneezing to wheezing to cramps, and ultimately to the most severe type of a reaction, which is called anaphylaxis. However, actually, we don't know precisely which one or which combination of molecules on the left mediate exactly the signs and symptoms that you can see here on the right. I will just circle one of these molecules, PAF, platelet activating factor, because I will refer to it in a few slides. Now, we said that there is a before and after, and the way that we've elected to visualize, to illustrate this idea is with the concept of the alpha, beta, and omega in food allergy. Here, alpha, the first letter of the Greek alphabet, is the origin, is the beginning. So it's the step of the process at which someone becomes allergic. So this is something that we immunologists call sensitization. And the hallmark of that sensitization is the production of IgE. Beta is the allergic reaction in itself, which we have just described a couple of slides ago. This is what immunologists refer to as the effector phase, and we know that all of these reactions are IgE mediated by and large. And lastly, there is omega, the last letter of the Greek alphabet. And by that, we refer to that phase of the process that becomes permanent, that becomes persistent, what we would call lifelong allergy, which is something that, as many of you know, occurs with certain types of foods, such as peanut, tree nuts, fish, and shellfish. We can understand that this alpha, beta, and omega is in fact a continuum process. Now, let's discuss in a little bit more detail the before, the alpha. How does one become allergic? Or how does one become sensitized? Well, actually, this is a question that is incredibly difficult to investigate in humans. And the reason is that sensitization, it's a silent process. So in other words, we know the consequences of sensitization, IgE, and we know that we can measure this. But what we don't know is when the process started. And that's the reason why, among others, that experimental models have been very useful to provide insights into the process of sensitization. So there it is. How can we make a mouse allergic to peanut? And why is that important? Well, it's important because mimicking food allergy and anaphylaxis in mice allows us to study what happens, where it happens, when it happens, how it happens, precisely. Now, a note about geography. Let's ask the question, where do we interact with allergens? Well, here in this diagram exhibits the various surfaces where we, our body, our immune system, interacts with external allergens. So in green, you can see that there is the nasal and tracheal and respiratory mucosa. Very important for interaction, of course, with aeroallergens. And here in red, you can see the oral and gastric and intestinal mucosa. Very important, naturally, in the case of food allergens. But in addition, here in blue, you can see the skin. So in other words, mucosal sites, as well as the skin, are the sites of interaction of us, our body, and allergens. Now, because we are speaking about food allergens, is intuitive to think that the way that we can make mice allergic to peanut is by using or developing a model of oral sensitization. You see, this is what we do. We take peanut and we feed mice with peanut along with something that is called cholerotoxin. Cholerotoxin is an adjuvant. It's something that will activate the immune system. And we do this typically 4 times, separated by 1 week each. And the reason we do this is because this allows us to generate a robust IgE response that is specific against peanut. And then a number of weeks later, we can challenge these mice, and then they undergo anaphylaxis. This type of experimental system, or variations of it, variations in the frequency or in the type of toxin that is used, have been used by many investigators. But this is oral sensitization. Can sensitization occur through a different route? And the answer is yes. Gideon Lack's group published this seminal paper in the New England Journal of Medicine in 2003. It's a retrospective study, but they studied thousands and thousands of children. And what they demonstrated was that peanut allergy was independently associated with the presence of rashes over joints and skin creases, and on oozing crusted rashes. And they also show that peanut allergy was associated with the use of skin preparations containing peanut oil. And that led to the authors to conclude that sensitization to peanut protein may occur in children through the application of peanut oil to inflamed skin. So that led to many investigators, including ourselves, to establish models, experimental models of skin sensitization to peanut. This is one such example. So what we do here is we apply peanut on a skin of the mouse that has been tape stripped. And this is done daily for about 10 days. Again, this is able to induce an immunoglobulin peanut-specific response, and a number of weeks later, we can challenge these mice, and these mice develop anaphylaxis. So this is a sort of the back alley of experimental models. What have we learned? Well, we've learned a great deal about the cells and molecules involved in the development of allergy. Very importantly, we've learned that oral administration of peanut alone induces tolerance, not allergy. We've also learned that the application of peanut on an intact, i.e. healthy skin, does not induce allergy. Then, as a counterpoint, we have learned that the development of food allergy in fact requires a local disturbance. In the case of oral exposure, this disturbance is accomplished by the administration of a toxin, which is something that may occur in the context, for example, of an infection. In the case of skin exposure, the disturbance is accomplished by causing epidermal injury. So in other words, inflaming skin, and that would be akin to eczema in children. So the bottom line is this. As a result of these local disturbances, the immune system sees food allergens as dangerous as opposed to harmless, not harmful, and then consequently initiates an allergic immune response. So you see, it's not simply a question of being exposed to the allergen, in this case, peanut. It's a question of the conditions on which the immune system sees that allergen, that peanut. Now, let's discuss now about the after, the omega of that entire process. Well, as mentioned, certain food allergies are lifelong in the majority of people. Majority means 80%. And this is in contrast to allergies to milk and egg or wheat that in fact are outgrown by the teenage years by about 80% as well. So it's really the mirror image compared to peanut and tree nuts and fish, etc. My colleague, Dr. Wasserman, and I have been very interested in our laboratory to investigate the mechanisms that underlie persistence of peanut allergy. And again, for a variety of reasons, this is a question that it's exceedingly difficult, if not impossible, to address in the humans. So we have used, again, experimental models. Now, we initiated a project approximately 5 years ago that was led by some very talented young investigators. But before we go into that project, we need another geographical node. And here the question is, where are IgE's positive cells generated? Well, the geography that we need is this. First of all, we need to have an awareness and understanding of the lymphatic system. So imagine that the circulatory system, the collection of arteries and veins, are the main highway. You know, here in Ontario with the Queen Elizabeth Way, for example. Well, we know that there is a parallel system of roads, which are the service roads. So we can imagine the lymphatic system as a parallel system to the circulatory one, but which is connected at certain points. If we look in closer detail at the lymphatic system, we see that throughout there are these areas here that you see in red in this picture that are the lymph nodes. Well, here, right here, this is where immune responses are generated. So note that, for example, if you have an upper respiratory infection, oftentimes you can feel that the nodes are swollen in your neck. So this is because an immune response is generated. Let us now take a look at a lymph node. Well, here, what you can see is that the allergen is transported from either the skin or the gastrointestinal mucosa into lymph nodes. And then there are a series of very sophisticated and complex interactions with different types of cells. Now, what is important is that the outcome of these interactions is the emergence of a cell that is called a B cell that expresses IgE. Now, the question then is, what is the fate of these IgE positive B cells? Well, on the one hand, they can differentiate into another cell that is called plasma cell. It has another name, very appropriately, which is antibody secreting cell. So this cell will exit the lymph nodes, go into the circulation, home in the bone marrow, and once it's sitting there, will start pumping IgE. So this IgE will go back into the circulation, percolate through all of the tissues, and coat mast cells or basophils in the circulation, as we described at the beginning. But then there is also another pathway. And this other pathway is that IgE positive B cells generated in the lymph node may become memory cells. These memory cells will also exit the lymph nodes, go into the circulation, and reside in peripheral tissues, such as the gut and others. These cells will do nothing, just sitting there, will consume very little energy, but they retain an important property, and that is the capacity to recognize the allergen if they see it again. Now, the project that we initiated in our laboratory lasted for about 5 years. And the result was the publication of this paper in the Journal of Allergy and Clinical Immunology on February 2017. We think it's a foundational paper because it provides very critical insights into the factors that underlie the persistence of food allergy, and specifically peanut allergy. It seems that it has been very well received by our scientific colleagues, because from February to August 2017, it has been downloaded an astonishing 1,300 times. Well, what is it that we discovered? Let me give you some perspective first. The lifespan of a laboratory mouse is 2 years. And the experiments that we conducted lasted, in a number of instances, up to 17 months. That's one of the reasons why it took so long for us to complete this project. In essence, what we did is we made mice allergic, and then we challenged these mice at a variety of points in time, understanding that between making mice allergic and the challenge, these mice were never ever exposed to peanut. The first and most important observation that we made was that mice actually remain reactive to peanut for virtually a lifetime. So in other words, we could make a mouse allergic to peanut at 6 weeks of age, and 17 months later, these mouse retain the capacity to react to peanut and developed an anaphylactic reaction. We had two fundamental questions. First, what persists of all of these factors that we know might play a role? Well, it's not IgE, because the half-life of IgE is 24 hours in the circulation, which means that in 48 hours it's gone. It's not that IgE remain bound to the mast cell for a lifetime, because it remained bound only for about 100 days. It's not that the cells that actually make IgE, plasma cells, were long-lived, because they only lasted 2 months. And it's not that the machinery that makes IgE positive B cells continues working forever. Actually, the only cell that remains virtually for the lifetime of the mouse is the memory cell, the memory B cell that retains the capacity to recognize and react to the allergen essentially forever. The second question that we ask is, how does it work? So what happens when an allergic mouse is re-exposed to peanut many months after? Well, here it is. So we know that the only cell that persists is the memory B cell. When peanut comes along again, that cell then will differentiate into a plasma cell. This is the cell that makes IgE. So really the reservoir of allergy is retained on that memory cell. We also know that that process is mediated by some particular molecules here that are referred to as interleukin-4 and interleukin-13. And we also have some insights that we reported on that paper about some of the cells that are the source of these cytokines. It is evident that if we are able to inhibit, prevent, neutralize the effect of these cytokines, we'll be able to prevent the transition and ultimately the production of IgE. So here, it's a very clear potential novel strategy. Now, of course, the next step is translation into humans. But let me tell you, this is much closer than what you may think. With all of this knowledge, we may now briefly consider different points of intervention. So for example, here, the LEAP study, which consists on early administration of peanut in very young infants, the LEAP study, is one such intervention. And this intervention, in fact, is a typical, prototypical, actually, prevention strategy. So this is not really applied to people with established allergy. You know, this is applied to people with the idea that it will prevent the development of allergy. I'm not going to go into any detail on this very important strategy, because that was an issue that was dealt with exquisite detail by Dr. Julia Upton on a previous seminar. Other points of intervention would be immediately before or immediately after the allergic reaction. So a potential strategy would be the use of drugs that neutralize or inhibit the effect of PAF, platelet activating factor. There's some data that suggests that this molecule may play a significant role in the generation of an allergic reaction. We'll see. PAF is not the only molecule. Remains to be seen how and when and to whom will be administered, and we'll have to await results to make a proper estimate of these potential treatment. Of course, then there is epinephrine, but epinephrine is only administered after the fact. But clearly, you can see already that whether is an intervention that is administered before an allergic reaction or after an allergic reaction, this in no way will affect the basis of food allergy. So this individual will continue being allergic to foods. And lastly, there is, of course, oral immunotherapy. And this is a strategy that has been investigated in great detail. Now, oral immunotherapy is part of a larger strategy that is referred to as allergen immunotherapy. And that means that allergen immunotherapy includes oral immunotherapy, sublingual immunotherapy, epicutaneous immunotherapy. Immunotherapy, in essence, consists of repeated administration of gradually increasing doses of allergen to individuals who are allergic to this particular allergen. It has been used for more than 100 years. And there's been many studies that have been conducted. So how can we make sense of all of this data? Well, fortunately, just a few months ago in Allergy, top European journal in allergy, on allergy, this paper was published, Allergen Immunotherapy for IgE-mediated food allergy, a systematic review and meta-analysis. This study is particularly important because meta-analysis is a methodology that provides the strongest evidence of quality. It's worth understanding that approach a little bit more. A meta-analysis is, in essence, a statistical analysis that combines multiple studies. And the idea is to test the pooled data to obtain or not the significance with a higher statistical power. So rather than looking at one study, you can look at 10 or 30 or 50 or whatever, including many more patients. A very important feature of a meta-analysis is that it selects individual studies based on a set of objective criteria, such that only methodologically sound studies are included. So this is the meta-analysis on allergen immunotherapy I was referring to. Here the authors identified over 1,800 studies. But you see, only 31, less than 2%, were selected. Collectively, these 31 studies included 1,300 patients. Of these 31 studies, OIT was used in 25, sublingual immunotherapy in 5, and epicutaneous immunotherapy in 1. The majority of the studies were for milk, 16, and egg, 11, and 7 studies for peanut. The main conclusion of this meta-analysis was that, well, the main benefit is that it is protective against accidental exposure. On the other hand, this study alerted us of a number of issues that we were aware of already. Immunotherapy is associated with a number of adverse effects. Compliance is difficult. It does not allow patients to eat the allergen ad lib. And if discontinued, sensitization comes back. So really the issue is not to determine whether immunotherapy is a good or bad strategy. It's an issue of understanding the advantages and the limitations of that treatment. So whether it's PAF or PAF-like strategies, or epinephrine, or OIT, what we can say with certainty is that none of these strategies are in fact a cure. Is there something new forthcoming? Well, what could be new? Well, what we are seeing already is different types of delivery approaches. So means and ways by which investigators can deliver the allergen, be it peanut or something else, in a very controlled fashion in pills, basically. We are also seeing different routes of delivery, allergen delivery. For example, the patch would be an example where the allergen is delivered to the skin, rather than orally. The issue with these various approaches is that the improvements may be technical, but not conceptual. Consider that the concept is still the same: repeated administration of gradually increasing doses of allergen. What else? Well, I think a combination therapy, which basically means immunotherapy, oral immunotherapy, or another form of immunotherapy plus something else, is gaining terrain. Here's one example: OIT plus anti-IgE, omalizumab. Omalizumab neutralizes IgE because it prevents the binding of IgE to the receptor that it's expressed on mast cells and basophils. In food allergy, we know that this combination indeed diminishes the adverse effects associated with immunotherapy, but as far as we can tell from these studies, it doesn't increase the efficacy of the immunotherapy in itself. And then perhaps the most novel strategies, and you can pick it up from the comments that I made before with respect to IL-4 and IL-13, another concept would be oral immunotherapy plus immune deviation. So that would mean the administration of a drug or an antibody that is able to generate an immune response that is actually protective. In summary, most allergic reactions to foods are mediated by IgE. IgE is produced by cells called plasma cells. These IgE-producing plasma cells are not long-lived. And in contrast, allergic memory B cells are. And probably these are the cells that allow certain types of food allergies to be persistent. Current therapies are beneficial to control symptoms, but disabling the allergic memory cell is the kind of a strategy that may transform the treatment of food allergy and perhaps lead to a cure. Thanks very much for your attention.
Thanks, Dr. Jordana, for a really interesting talk. A lot of people, when they registered with us, they had questions about a cure. We were having more questions come in through the webinar as well, and they're interested in understanding a bit more about when a cure will, be found. One of the things that we asked Dr. Jordana to do, because we had so many questions about this beforehand, is we asked him to put together one more slide and to talk about some key concepts and how we might want to think about the term cure. So if you could switch over to your next slide, Dr. Jordana, and go over that, that would be wonderful.
Thank you, Lori. Indeed, there were quite a number of questions related to the concept of a cure. Well, cure it's a simple word, but it's a concept that cannot be used lightly. Let me use some analogies. Suppose that someone has a respiratory infection, even a pneumonia, and then the doctor prescribes the correct antibiotic that kills the germs that are causing that infection. And eventually, after a few weeks, the person recovers fully. We would consider this to be a cure. Now, consider now someone with asthma. We have very excellent inhalers available to our patients. And these inhalers are able to control the symptoms of asthma in the majority of patients. But we know full well that these inhalers do not cure asthma. but they do, is to control the symptoms very well. So what could be a cure in food allergy? In a strict sense, what we would say is that a cure would be a treatment that generates tolerance such that exposure to a food allergen after the treatment is finished does not cause an allergic reaction under any circumstances. It's a very strict definition. And what we think is that because none of the treatments that are at the moment available actually are a cure, we think that these may be accomplished by, I wish to reiterate, disabling the machinery that makes IgE. And then conceptually, there would be at least two strategies. One would be by deleting the cells that retained the memory, or strategies that are able to prevent that transition, from memory cell to IgE producing cells. That would be a strategy. Another strategy would be by regulating allergic memory cells through a process of suppression. So here on the one hand, you've got the allergic memory cell, but you could imagine, and a number of people are working on this idea, that you may want to generate another cell which main ability is to suppress the allergic cell. So these are different conceptual approaches that I think go to the heart of the problem in food allergy, which is, the disabling of the machinery that makes IgE.
Thanks, Dr. Jordana. We've had some other questions come in. The next one is about combination therapy. You mentioned that perhaps one of the things that, or some of the things that are being looked at is peanut oral immunotherapy plus something else, for example. Could you talk a bit more about that?
So one of the strategies that has been proposed that would follow within that concept of combination therapy is the use of probiotics. And so this is in reference to the Australian study that was recently published in the Lancet Child and Adolescent Health journal. The article and news releases by the Institute, the study was conducted, as well as a number of media outlets, led to much hype, including statements such as, "This is the strongest evidence yet that a cure may be possible for peanut allergy." This conclusion is grossly misleading. Now, for the audience, I would suggest a few resources. Dr. Wasserman, my colleague Dr. Wasserman, and I, as well as Dr. Derek Chu, published a statement on this particular study on the Food Allergy Canada website. It was also published on the Allergen NCE website. Furthermore, we collaborated with Gwen Smith on a piece that was published on Allergy Living magazine. And lastly, a letter to the editor of the Lancet Child and Adolescent Health has been very recently published by us and our collaborator, Dr. Wayne Schreffler from the Massachusetts General Hospital. So these are resources where we have elaborated in detail about this study. In essence, this study is a follow-up to another study called the Parent Study, in which the authors investigated the impact of a combined treatment, oral immunotherapy plus probiotics versus, and attention, because this is a very important point, versus placebo. So patients who completed the parent study were contacted 4 years later, and the primary outcome was how many of the patients in the active treatment, OIT plus probiotics, were still eating peanut. Well, first, the claim that most patients treated with probiotics plus immunotherapy were cured is false. Patients that were quote unquote cured had the same number of food allergic reactions and the same skin prick test positivity as those that never received any immunotherapy. Secondly, the claim that probiotics were responsible for any efficacy is unproven. In the parent study, there were only 2 groups, those that received a combination of peanut and probiotics versus those that received placebo. There was no group that received peanut without probiotics or probiotics alone to determine the contribution, if any, that the probiotic made to any of those outcomes. And thirdly, we know perfectly well that about 60 to 70% of people subjected to oral immunotherapy will become desensitized. So it's absolutely no surprise and certainly not a breakthrough, as it was claimed that those people, those individuals, those patients who continued eating peanut, 60 to 70% would remain desensitized, because that's consistent with all the literature that it's available out there. There are a number of other methodological issues, but I encourage you to go to the resources that I've described for a more detailed analysis.
So the next question on that comes in terms of allergies that persist and are lifelong. You mentioned that the ones that seem to be lifelong are things like peanuts, tree nuts, shellfish, etc. And others are sometimes outgrown, and you mentioned egg, milk, and wheat. Can you talk about why that happens? Like, why do some persist and others might resolve?
That is a very important question, but it's also a very difficult one, and I think that, to be completely honest with you, we really don't have an answer as to why some allergens are associated with lifelong allergy, whereas others they're not. But perhaps we can address the question indirectly. And the immunogenicity, so in other words, the potential of a food or a protein to cause an allergic reaction varies. We know that. And it's important to realize that the occurrence of an allergic reaction actually depends on a complex interplay between the cells of the immune system and the protein in itself, the purported allergen. So how can we explain these differences, this variability in immunogenicity? Well, there are structural and biochemical differences among allergens that may influence the way that the immune system captures and processes the allergen in different ways. We also know that many of the important allergens, allergenic allergens really, are exceptionally heat stable and retain immunogenicity after heating. We also know that most of the common allergenic proteins are relatively stable in gastric fluid, whereas minimally allergenic proteins are labile. In other words, are degraded very easily. This is important because when degraded, these proteins are unlikely to persist in the gastrointestinal tract for sufficient time to be captured for immune cells. There are also differences in terms of a process that it's called glycosylation. So in other words, the number of sugars sugar molecules that are present in that allergen. And this again influences the rate of uptake of these allergens by cells of the immune system. So these are different reasons that may contribute to different immunogenicity, to different allergenicities, to how allergenic these proteins might be. One of the things that it's important is that certainly all of those things are going to influence the way that the immune response is going to be generated. So, and that may influence ultimately the persistence of the reaction. It's important finally to note that a lot of these structural and biochemical features that I've mentioned can be influenced by the manufacturing or the processing processes. So for example, we already know that baked milk is less immunogenic than raw milk. We also know that, for example, in Southeast Asia, people consume huge amounts of peanut, and yet peanut allergy is not a problem. There may be different reasons for that. Maybe that people are exposed much early to peanut, but also there may be other reasons, such as that in those countries, peanuts are consumed largely steamed, boiled, or raw, whereas in the West, peanuts are consumed basically roasted, and we know that roasting makes a peanut more allergenic. I think it's interesting because a person asked, well, you can be allergic to soybeans, to soy, but you still can eat or ingest peanut oil without having a reaction. So I think that this is another example of the influence of processing. And the reason is that in order to make soya oil, this requires very, very high temperatures. And when very high temperatures are used, I think that the structure, the protein structure of soya is degraded to a point that it becomes non-immunogenic. So there's lots of different reasons that can contribute to the immunogenicity and perhaps the persistence of this.
Okay, so Dr. Jordana, one last question and maybe a quick response, and then we've got to wrap up. So we've had several questions about how far are we from something that's commercially available, and I'll read this one. It says, given positive results for OIT from the meta-analysis, how far away do you think we are to a commercially available OIT program? And with that, maybe what you could shed some light on is the process for bringing something to market.
My impression, and the impression of many of my colleagues and clinical colleagues as well, is that if we talk about immunotherapy alone as a treatment, that may have reached its ceiling in terms of efficacy. So I believe that the future is on combination therapy. So in other words, oral immunotherapy along with another drug, antibody, product that is able to, in some fundamental way, deviate the immune system. So as I was talking earlier about how peanut allergy is persistent, the importance of memory cells, and certain types of interleukins, 4 and 13, in that process. Well, I can tell you, this is not far away. And in a few months, not years, in a few months, studies following exactly that concept are going to be initiated. It's going to take years because a process needs to be followed. So first of all, efficacy needs to be proven in some pilot studies, but then multi-center type of studies are required, etc., etc. So even if those type of studies are initiated in just a few months, as they will, it's going to take 3, 4, maybe 5 years until we have a clear idea as to what the benefit of these novel interventions might bring.
Thanks very much, Dr. Jordana. We'd like to take a moment to thank our sponsors for our ongoing webinar series, Pfizer Canada, the Sean Delaney Memorial Golf Classic, the Walter and Maria Schroeder Foundation, and the Peanut Bureau of Canada, to name a few. Please consider also donating to Food Allergy Canada. We're a nationally registered charity and we rely on donations in order to provide these services to support the food allergy community, such as these informative webinars. We'd be grateful for your support. You can find the donation button on foodallergycanada.ca/donate, and we'd greatly appreciate your contribution. If you'd like to see this webinar again, it will be archived in the near future, and we'll be getting the word out to you where you can find that link. However, if you'd like to go back and see some of our other webinars that we spoke about, go to our website at foodallergycanada.ca, and under resources and recorded webinars, you'll find them there. I thank you all on behalf of the team at Food Allergy Canada for your participation today. We hope that you found the information useful and helpful. There's going to be a short survey that we're going to be sending you. We'd greatly appreciate your doing it quickly because we like to hear what you found useful and what else you'd like to hear in future. Speaking of the future, as Dr. Jordana alluded to, it's quite complex trying to find something that's going to help us, whether it's to soften the response of an allergic reaction through a therapy, or one day, hopefully, get rid of the allergy when we'll have a cure, and that day is not right around the corner. However, there's a lot of things that we can do to live confidently and safely. While progress is being made in the research field, we encourage you to go back to our website and find information there. They're all medically reviewed and they're designed for you or your patients. Thank you very much for your time today, and again, we hope you enjoyed today's session.